Good morning, everybody. Like I say, my name is Amanda Grimm. I'm the Senior Segment Marketing Manager for our antibody drug discovery portfolio, as well as IVD. At GenScript's presentation today is Seamless Concept to Candidate, talking about our cutting-edge technologies for your bispecific and antibody development. So the contents of my presentation today will include a little bit of information about GenScript. I'll touch on some of the bispecific antibody trends, as well as introduce some of our antibody solutions, and then I'll close out with some information for your consideration if you're looking for partnership or support in your work. Okay, so starting with some information about GenScript. If you're not familiar with us, or this is new information, we do have over 20 years of development experience. Over 18 years of that is specific to antibody service. To date, we have completed over 150,000 antibody projects. And then when we look at the history milestones of the GenScript group, GenScript was founded in Piscataway, New Jersey, in US, which is where I'm based out of. We started with gene synthesis, and since then we've added a variety of different services to our molecular biology portfolio, adding the antibody service, protein expression, as well as many others. We've also added additional subsidiaries since then. We have Bestzyme, Legend Biotech, as well as ProBio. And then just quickly noting a huge milestone was our collaboration between Legend Biotech and Janssen to bring a cell therapy product to market in 2022. Just a little bit more information about the individual subsidiaries. We have the Life Science CRO. Again, that's where I'm based out of. We have ProBio, our biologics CDMO, who's also sharing a booth space with us today. I mentioned Legend Biotech, our cell therapy subsidiary, and then also Bestzyme, covering our synthetic biology services. Okay. I'll go ahead and touch on some trends with bispecifics, by no means as in-depth as Dr. Klein went into this morning. But bispecifics really have provided a revolutionary attempt with therapeutics, and market reports in this case showing potentially reaching over $66 billion by 2031. When we look at the approvals as of 2023, the majority of these have really shown great results for patients with cancer indications. We have a smaller subset, like say for inflammatory and autoimmune disorder, and then we have a smaller other group covering things like ophthalmology. These have really been revolutionary because they're able to address novel mechanisms of action. They provide a more targeted and efficient therapeutic outcome, and this is all made possible due to a lot of these new technologies that are allowing for the development of these molecules. It's not to say that there's not other development challenges associated with that. Again, I think Dr. Klein's presentation really covered a lot more of that. But essentially looking at the various associated indications, understanding the disease itself, identifying targets that are have a high affinity and specific, and that are also going to be dually complementary to the mechanism of action that it's intended for. And so with that, I'll move to introducing GenScript solutions for antibody drug development. As I move into this new section, it's meant to be more informative in talking about a number of the core services that we have available, as well as tools to support that seamless transition from your concept to market. So I'll start first with an image of the antibody drug discovery workflow. The image is really based on the traditional mouse hybridoma platform. The intent here is really to give a visual to divide the workflow into four stages, being target identification, lead generation and optimization, preclinical and clinical development. This slide is to expand on those four stages to kind of give you an idea of the coverage that GenScript has at any stage that you're in. This is not all-encompassing of everything that we offer. It's just to kind of give you the impression that we have a lot of coverage. Okay, so the first solution I want to start with is to talk about codon optimization and plasmid design as a way to maximize your chances to achieve those functional proteins. We have a free tool that uses smart error checking as well as smart algorithm in order to achieve this. You may already be using, or maybe you're familiar with our GenSmart platform. We did upgrade this to a 2.0 version much earlier in the year. But when we look at the codon optimization component of this, it now utilizes the population immune algorithm, which is going to consider over 200 factors like the GC content, codon usage, and mRNA destabilizing motifs. It also is now applicable to over 50 host proteins, including CHO, E. coli, T cell, and even some plant species. We also have a free construct design tool as part of this platform where you can optimize your wild type or recombinant sequences depending on the expression system. And then it also includes over 200 commonly used commercial vectors and over 2,000 commonly used functional parts. A little bit of data just to emphasize, what codon optimization can do for your proteins. This is some internal data looking at 3 genes for each of GFP and JNK3 proteins. In the Western blot analysis, we can really see that visual difference between the wild type and an optimized sequence for both protein examples. And then whenever we chart that in the graph, we can see, visually, and for each protein, we're getting about 18-fold with the GFP and about 8-fold with the JNK. Next, I want to talk about expression-ready plasmids. We do have in our database 2 free plasmid backbones if people are working on iterations of their antibody candidates. This service was specifically developed for our antibody discovery customers, and it's going to address some of those core features. This is our antibody gene to plasmid package. It's part of our new Flash gene service that was launched earlier this year. We've been doing a lot of launching and upgrading this year. But essentially, you can see with these 2 vector backbones that we're going to be addressing those key areas that I mentioned, like the variable heavy and light chain, optimizing promoters, signal peptide, optimizing for linker peptides in the case of bispecifics, as well as providing choices for purification tags for your downstream purification. One of the things to note with the Flash gene service in general is that this is bundling some of these key manual I can't think of the word, I'm sorry. But essentially, we're bundling the gene synthesis, the vector cloning, and plasmid prep into one service so that by the time you receive the plasmids, these are ready for you to use in your experiments. Next, I'm going to move into the recombinant antibody and protein expression system. This is a platform that's going to provide you with easy scale-up, high-quality protein products, and provide that fast delivery to keep you on track with your projects and milestones. It's our proprietary TurboCho expression platform. Like say, it is a scale-up platform, so no matter where you're at in the stage of discovery, we have an option for that. So for example, when we look at the high throughput, this is going to be transient expression, providing you with smaller protein yields, but a very fast turnaround of 5 business days. As you move through the process, scaling up to our Express platform, still transient, but larger volumes, but also you're getting more deliverables, including like purity and endotoxin levels. And then as you move further into the later stages, transitioning to the high performance option, this is going to be your stable protein expression with those gram to kilogram volumes. And since we're talking about bispecific engineering and development, I really want to highlight our Express version of this platform. This is going to be the platform that we use for our bispecific expression. We have a team of scientists that have the capabilities to offer bispecific design of your projects. That includes over 10 years bispecific development experience, and they've completed over 5,000 bispecific projects to date. The capabilities that are addressed in this include a specialized format design, using our AI-driven codon optimization, optimizing the production process as well as the plasmid ratio, and then also choosing from multiple purification column options. The image over here on the right, looking at some of the bispecific achievements, looking at those different groups of bispecifics, of formats, symmetric and asymmetric. The purpose here is really to kind of call out that we have an entire web page dedicated to these over 70 formats that we have produced. You can interact with each one individually and learn a little bit more about the manufacturability and the purification involved with each of those. And then here I just want to expand a little bit more in each of those bispecific format groups, addressing general production challenges associated with each of those groups, that those are also things that we're going to be addressing, as I mentioned, heavy light chain, aggregation, cleavage, as well as folding. And then I did want to provide just a little bit of data to highlight one of these bispecifics, similar data that you would see on the website. In this case, we have an IgG-like bispecific using the cross-map technology with that CH1Cl. And then the purification that we were able to achieve with this specific format was 374 milligrams per liter. And then I did just include some of the images highlighting purification of the SDS page. We have the heavy chain at 65 kilodaltons and light chain at, I think that's 25. And then when we look at it under non-reducing conditions, we can see a very clear band of the whole molecule. And then, of course, over here we have the size exclusion chromatography graph. Okay, next, improving sensitivity and specificity of your candidate leads. Mutant libraries are a great way to do that. So we also include a semiconductor-based oligosynthesis platform. This is going to generate a superior library density and also provide precise control over the synthesis of those oligo strands. Our precision mutant libraries are a great way to address the CDR engineering as well as improving ligand receptor binding. This technology is going to allow us to provide even distribution of amino acids with very little bias. It's going to include user-defined codons, and we're also able to eliminate the formation of stop codons with this. We're able to provide saturation scanning, site saturation, as well as combinatorial. Essentially what we are going to do is generate those oligo strands on a semiconductor chip. Those strands are then ligated into plasmids, sequenced, and then sent to you to perform additional screening assays, whether that's phage display or cell surface expression. I do have some data comparing our semiconductor generated precision mutant library compared to a traditional NNK library. This is data I pulled from our website. We have a pretty in-depth application note that talks way more in detail than this, but I really just wanted to highlight that difference, in this case being a combinatorial mutagenesis library. I didn't get this edited, but essentially this is looking at 4 sequence mutations. And so in this first figure here, we can see in the degenerate library versus the precision mutant that the precision mutant has a lot better distribution when it comes to the amino acid sequence. And then whenever we look at the screening efficiency, again, the precision mutant library is much higher, whereas in the NNK, much less efficient due to those truncated unwanted sequences due to the stop codons. All right, next we'll move into developability. We want to know whether our antibodies are going to be manufacturable, are they going to be immunogenic, what's the viscosity, aggregation, and degradation look like. And we do have an AI-based tool in order to do that. This is kind of introducing you to that immunoinformatic platform that we use for our developability. It's also a platform that's integrated across a number of different places along the workflow. But specifically, because we're talking about developability, this is the section that I'm referring to. In addition to the things I mentioned in the first slide, this area is also going to provide prediction for post-translational modifications as well as bispecific structure annotation. I'm not going to go into detail on this, but if you are interested in learning more about this platform and how we're utilizing it, you can screenshot that QR code and you can hear directly from our bioinformatics scientist, Dr. Carter Howe. I will also, before moving to the next slide, note that this developability assessment is integrated now as of the summer as part of the TurboCho Smart Quote process. So all you have to do during the order for that Smart Quote is select that you want the report, and it's going to give you some information addressing these. And again, because we're talking about bispecifics this morning, I do want to just provide a little snapshot of that bispecific structure annotation from the developability report. And essentially what we're doing here is you're submitting your sequence, and it's going to be compared against nearly 400 formats in our database. The sequence will be broken up into fragments, but essentially what the readout is going to show for bispecific in test case 1 is the sequence mode is going to report in this format, is going to provide you a class, a subclass, in this case Fab diabody. It's going to give you the chain ratio as well as a reference structure. Now if you have a sequence that is not bispecific and that's entered into here, it's also going to recognize that it's not bispecific, and it's going to report similarly in this test case 2, where it's not matched, it'll provide an output of a similar class. In this case, it recognized it as an antibody fragment, subclass, single chain variant fragment, as well as provide you with a potential structure for that. Okay, next we'll move on to humanization. This is also a feature of that immunoinformatic platform. Thus the AI evaluation. But essentially, humanization is going to be important in order to help you increase the efficacy of your antibody as well as reduce immunogenicity and help with the likelihood of that candidate making it to market. This is what that workflow would look like, essentially providing us with the sequence. That sequence is then compared within our database, we come up with 15 humanized and wild-type antibodies, where then we do some additional experiments on our end using ELISA binding and EC50 ranking in order to identify those top 5. And then those would be provided to you in a small-scale transfection for you to continue your experiments. I included some icons here of cats and dogs, mainly to remind me to tell you that if you are working in veterinary therapeutics, we also have the capability to felinize and caninize antibodies as well. Okay, and the last area of solutions I want to share with you related to the safety and efficacy of your clinical development, providing you with tools for pharmacokinetics and anti-drug antibodies. So that would be the anti-idiotype antibody service that we provide. When you take advantage of the MonoRab platform, you're going to get high quality, highly specific antibodies to use in your PK and immunogenicity assays. We're able to develop blocking antibodies, non-blocking, drug target complex specific antibodies, and in order to do this, we can use a number of different immunogens to do that, including peptides, DNA, RNA, bispecifics, and more. A little bit of data related to the anti-ID generation. In this specific example, we had a project target for a trispecific antibody. The immunogen we ended up using was a single-chain variable fragment conjugated to His. And then the goal here was to generate 4 blocking and 1 non-blocking anti-ID antibody. The results here were that we were able to generate 133 clones. We found that 96 of those have a blocking rate of over 30%. And then the top 3 blocking clones were niched down further from that. The data really is just to demonstrate, in this case here, we did also immunize mice at the same time to do a comparison, and the result was really that we got a much better outcome with the rabbit, as well as achieving more clones than we did with the mouse. And then the last little piece of data just showing those 3 clones that I mentioned here, graphing them with the OD against concentration. Okay, and then the last piece of data to share with you is a summary of ADA polyclonal antibodies, because these are important extending into your clinical development as well as post-market monitoring. Again, highlighting our capabilities to use a variety of different antigens to do this. Bispecific here, where we were able to generate 56.8 milligrams of total ADA with a purity of 97%, very high ELISA titer with the cross-absorption of 0.4. Okay, that brings me to kind of the closing section of what may be of interest if you're considering partnership or looking for support in your projects. Again, we have over 20 years of experience, 20 years of partnerships. This year, we reached a milestone of over 100,000 successful customer research publications in peer-reviewed journals, notably in Cell, Nature, and PNAS. And then this is just an overview of what a project may look like with us, that project initiation, discussions, agreements, identifying the project scope, timelines, technical support involved, and then of course we provide you with the deliverables with high-quality products and ongoing support. And with that, I thank you for your time. If you haven't been to the booth yet, we are 1218 on the lower level, back by the coffee and food. We also have a poster by Dr. Soon Gao on our DNA mutant library, so if you have questions more specific to the DNA libraries, mutant libraries, she's a great resource for that. And then in case you did miss the presentation yesterday, we had a sponsored talk with Dr. Field at Lab Genius talking about his research and work. So if you have questions, I would encourage you to come by the booth. I'm happy to talk one-on-one. We do have some of our scientists and product managers here available as well, who can give you more of those technical details related to the antibody services. So thank you.
Thank you very much. Do we have questions? I would have one indeed. You mentioned your turbo CHO expression. What's the throughput? Let's assume I want to screen bispecifics and need hundreds of different molecules. Yeah. Would you be able to produce those?
I want to say yes, but I would definitely need to check with the product manager on that.
Mm-hmm. Okay, and a follow-up question on that. You mentioned your humanization services. Does this only relates to IgG antibodies, or does it also relates to camelid-derived VHHs and so on?
It should be applicable to both, yeah.
Okay. Yeah. All right. Any other question? If not, then thank you very much.
Thank you.